Endospore-forming bacteria consist of two bacterial families, the Bacillus bacteria, and we already discussed anthrax, which is caused by Bacillus anthracis, and now we are left with the Clostridial bacteria. Clostridia are strict anaerobic bacteria. That means they cannot grow in an atmosphere that contains more than about 0.5% of oxygen. In order to survive, they then form spores. And there are about 150 known different species of Clostridia, but most of them live as saprophytes in the soil and on decaying vegetation. They also occur in the water, and they are of no veterinary or medical significance. But a few species are pathogens, either obligate pathogens or opportunistic pathogens, and they can occur naturally in the intestines of animals or in the environment. And we are going to focus on those Clostridia of veterinary importance. In healthy animals, both the small and the large intestine contain Clostridia. And if the animal dies, those Clostridia start invading the abdominal organs from about 6 hours after death. That means we must be very careful not to misdiagnose the presence of Clostridia in those tissues as the cause of death, while it is rather a consequence of death. Which species of Clostridia are of veterinary importance? There are three main groups which can cause very different disease syndromes. Firstly, the Clostridium perfringens group, which mainly causes enteric diseases. The second one is the histotoxic Clostridium group. They cause muscular disease, specifically in the form of gas gangrene. And thirdly, the neurotoxic Clostridia. These are the causes of tetanus and botulism. All pathogenic Clostridia have in common that they produce very potent exotoxins with a variety of effects. Some of these toxins have a very localized effect, like for example necrosis, or systemic effects, like enterotoxemias. They all have in common that the disease can have a very rapid progression, as in peracute, and leading to sudden death. Within the Clostridium perfringens group, the most important toxins are alpha, beta, and epsilon toxins. And these Clostridium perfringens types are determined based on the presence or absence of these different toxins. Alpha toxin has a very localized effect, as it first destabilizes membranes and connective tissues, and later leads to necrosis on exactly that spot. Beta toxin also causes necrosis, but on a much broader scale, while epsilon toxin causes an increased capillary permeability, which leads to leaking of capillaries. Clostridial exotoxins can be inactivated to produce very effective toxoid vaccines. Those vaccines stimulate an immune response which protects the animal from the effects of the toxin, but does not inhibit the growth of the organism itself. Those vaccine formulations are available either for individual diseases or in combination with other clostridial vaccines. The Clostridium perfringens group of bacteria cause two groups of diseases: wound infections and enterotoxemias. Wound infections are more important in humans than in animals, but the list of enterotoxemias in different animals is very long, and not all are of equal importance. Really important are the ones in red, and we will focus on those diseases for this presentation. The first important disease is lamb dysentery, or Clostridium perfringens type B infection. It affects newborn lambs and kids less than 14 days old, and in most cases it causes death after 2 to 4 days. First of all, please bear in mind that Clostridium perfringens type B is an obligate pathogen, which means it is not part of the normal flora in the gut. The lambs pick it up from feces containing the organism in a contaminated environment, mostly from the soiled teats of the dams during suckling. Please do not confuse it with a milk-borne infection, as for example in brucellosis, as the organism is not excreted in the milk. The risk of a buildup of the bacteria in the environment is obviously greatest in intensive sheep farming with crowding of sheep at night under unhygienic conditions. After ingestion and germination of the spores, they colonize in the ileum. While they replicate to very high numbers, they produce the beta toxin, which causes necrosis and ulcers in the intestinal wall. But the toxin is only effective in animals which are too young to produce trypsin, because trypsin will inactivate the beta toxin. So that means it explains why only lambs below the age of 3 weeks are affected by lamb dysentery. The action of the toxin also leads to increased motility of the gut and a disrupted absorption, and therefore leading to diarrhea. And due to the loss of fluids, there is dehydration, which in endemic farms, about a quarter of lambs can become infected and die. And if the lambs die peracutely, no clinical signs are seen as the disease progresses too fast. Animals that survive for a few days, they show abdominal pain and diarrhea and don't feed up to death. As we get to diagnosing the disease, the history of a previous occurrence of lamb dysentery on the farm gives a very strong indication, together with the age of the affected animals. But the disease can best be confirmed by culture, as the organism is an obligate bacterium, so it should not be present in the gut. We need to be careful of differential diagnosis affecting the same age group of lambs, namely colibacillosis and salmonellosis. The pillar of control is vaccination of pregnant ewes about a month before the lambs are born. In support to vaccination, the farmer should separate the affected animals to avoid the environmental contamination, and antibiotic treatment is a good support in early cases, but often too late in advanced cases. The second, and probably the most important Clostridial disease, is pulpy kidney. Pulpy kidney is caused by an enterotoxemia, meaning the toxin produced in the intestine is absorbed and distributed via the blood. In this way, the toxin can have far-reaching effect in multiple organs apart from the intestine. In the case of pulpy kidney, the toxin that is responsible is the epsilon toxin, which causes capillary permeability. What do we need to know about C. perfringens type D? Most important, that it is a saccharolytic organism, meaning it metabolizes carbohydrates and uses it as energy source. Secondly, it is not an obligate pathogen, but it occurs naturally in small to moderate numbers in the ileum, and also produces small amounts of toxins, but they are too small to cause an effect. And here comes the answer to when this organism acts as a pathogen. If there is a sudden change in diet to a much more energy-rich food, the rumen microflora is not able to digest the overload of carbohydrates. An undigested starch enters the small intestine, where the C. perfringens now indulge in these carbohydrates and multiply rapidly to extremely high numbers. The epsilon toxin is produced by the bacteria as a prototoxin, which must be activated to be effective. This is done by trypsin. So note that we are dealing with the opposite situation from C. perfringens type B, where trypsin inactivates the beta toxin. So let's apply our common sense to this. If trypsin is required to activate the prototoxin, pulpy kidney can only affect lambs which are older than 3 weeks. Due to the increased capillary permeability caused by the epsilon toxin in the intestine, the toxin can enter the bloodstream and affects the endothelium of blood vessels in the kidneys, leading to the pathological picture of the typical pulpy kidney, which gave the disease its name. Further, the lungs and brain can be affected by edema. In the brain, the so-called focal symmetrical encephalomalacia, or FSE, is caused by vascular insufficiency, basically a malfunctioning of the blood vessels to nourish the brain. This requires some days to develop and is hence only seen in animals that survive the peracute stage, and affected animals show neurological signs. In the forefront of management procedures, which could lead to pulpy kidney, are, for example, tail docking, castration, or individual marking, causing excessive amount of stress, pain, and followed by a stasis of the intestinal tract. Who is mostly affected by pulpy kidney? The disease is very common in sheep, but rather rare in cattle. In sheep, all age groups can be affected if a predisposing factor is present, but it occurs most commonly in young lambs, 3 weeks to 3 months old, so that coincides with weaning, and then the group of 6 to 12 months of age, which is associated with the time when the lambs enter feedlots and receive an energy-rich diet. Forms of the disease very common peracute, so death can occur within less than 2 hours. Most common is the acute form, anything up to about 12 hours, and we talk about a chronic form when the animals survive up to 7 days. And very often, the fattest sheep is the first to die. In other words, the sheep in the best condition are at highest risk, probably because they are just prone to eat the most food in the shortest possible time when they get a chance to. The clinical signs seen in the acute and chronic forms are listed in the following 2 slides and are self-explanatory. To confirm pulpy kidney, intestinal content can be submitted to the lab for culture. The best practice is to tie off a piece of small intestine at 2 points, about 20 centimeters apart, and remove this piece as a sample. Alternatively, an intestinal swab can be packaged under anaerobic conditions. To demonstrate the lesions in the brain of FSE or other organs, samples can be collected in formalin for histopathological examination. The most important tool to control pulpy kidney successfully is vaccination using the program as follows.